My name is Jeff Lowenfels, and I am author of a quadrilogy of books on the soil called Teeming series.
Ladies and gentlemen,
all of you freaks, mutation.
Curiouser and curiouser.
You're a prophet of decentralization.
To reestablish a cosmic context for mankind.
the search and striving for truth and knowledge.
There will be a degeneration of ideas.
What kind of place is this?
Hey everybody, this is Jason Wilson with Isn't Life Curious? Thanks so much for tuning in once again. Today I'm very excited to be catching up with an author and fellow avid gardener. I've been reading his books for many years. He's now got a quadrilogy of books all about plants and their interactions with soil. I'm here with Jeff Lowenfels. Jeff, thank you so much for being willing to come on the podcast today.
Pleasure to be here today. No question. You can hardly shut me up when it comes to this subject.
Absolutely, and it's an important subject. And before we dive into the book that you just wrote, Teeming with Bacteria, which is all about the rhizophagy cycle, and we'll dive into how exciting that is and why people should be excited about bacteria when they usually probably shudder at hearing the word. The first thing I wanted to ask you is, you're on your fourth book. Where does this passion come from that you have for plants and growing and trying to find better ways to do it?
Sure. Well, I've always been a gardener. My father was a big-time gardener. His father was a big-time gardener. Everything we did centered around gardening. And if you wanted to spend time with either of them, you did so in the garden. So I was a gardener at a very early age and have continued ever since. I moved to Anchorage, Alaska from New York. I live in Alaska right now. And I write a garden column up here, which I've been writing for some 47 or 8 years. They started out as letters to my father about what I was doing in the garden. And so that became the garden column. But so I've always had a need to garden because I had a need, of course, to be near my father. And it just becomes addictive if you learn enough about it. If you're just the kind of gardener who goes to the store, maybe at one of the big box stores, buys a 6-pack of marigolds and sticks them in a box or a planter someplace and waters them, you're not really a gardener. And what it might take for you is to understand a little bit more about what's going on in the soil and in the plant to stimulate you to become a gardener. And that's sort of what my books try to do. I am thoroughly convinced we all need to be organic in our gardening habits. And so my books are the science behind organic gardening. The first one, Teeming with Microbes, was written in 2006, and it set forth a vision of the soil food web presented by Dr. Elaine Ingham. The second one talked about how the nutrients which were released by the soil food web got into the plants and what they did once they got in there. That was Teeming with Nutrients. Mycorrhizal fungi, which were just a paragraph or so in the first Teeming with Microbes, became so important that they deserved their entire book. And so Teeming with Fungi came with that. And in 2010, there was a new discovery that indicated that bacteria, which we thought were simply eaten, the excess of those bacteria lunges were deposited in the soil. They migrated into the roots. Well, it turns out some of the bacteria don't get eaten by nematodes and protozoa, which eat most of the bacteria. They get eaten by the plant. Not really eaten. They move into the root cell of meristem root cells, of very young cells in roots. And there, the plant, which has attracted them to come inside, strips them of nutrients and multiplies them. They themselves escape the cell by creating root hairs from which they are shot out of. They regain their nutrients and then go back into the next set of meristem cells a couple of days later. So you've got this cycle going on. It was discovered by an Australian. Most people pronounce it, and I know I don't pronounce it the right way. Most people pronounce it Rhizophagy or Rhizophagy. I say Rhizophagy. I don't have an Australian accent, and so I can only spell it the way I see it. Root eating. And that's exactly what's going on. It's fascinating new science, and it's all now outlined in this book. The first book, Teaming with Microbes, was written for Dr. Elaine Ingham to explain the soil food web. This book is written for Dr. James White, who is a professor at Rutgers University, to explain this new addition to the soil food web. A lot of fun to write, and some of the implications of it are simply mind-blowing and useful.
Well, and that's I'm glad we're diving right in, because that's really what I wanted to get into, and you just painted a really perfect picture of what I say rhizophagy, this rhizophagy
and you're probably
cycle.
saying it right.
I always tell people, especially when it comes to science stuff, these are a lot of words that people all over the country write and read all the time, and we very rarely get together and say these words out loud. And then when we do, we're all saying different things.
Right.
But it's really an incredible dance that's happening between the plant and these bacteria, and all of these cycles that are going on, and the interesting mutualisms that occur. But then I think exactly, what you just pointed on, like the practical implications of understanding this cycle. I think that's what anybody who is hearing us talk about this, they might think, well, it's interesting, but what does it really mean for gardeners or even farming? And that's in our entire approach to trying to work with the soil. What are those implications?
Well, let's start with the generalized soil food web. We've learned that if we let that soil food web operate naturally, the plant will take care of itself. And so the biggest implication of the generalized soil food web is you don't rototill. That breaks up the fungal network. That puts the bacteria that are supposed to be in the root zone in a different location. And when you cut a worm in half, you don't get two worms. You get two dead halves of a worm. So we've also learned that if you apply nitrogen as a chemical to your soils, they go into the plant, but they cause the plant to shut down part of their systems, so that the plant no longer operates the same way, and you don't get the same benefits from in particular the bacteria that you would get. And that's an important thing to point out, that when these bacteria enter into the plant, they cause the plant to become a better plant. So there's two basic kinds. The first are these rhizophagy, or I like it the way you say it better, bacteria. They actually, when they go in and live in the root system, when they go in, they cause the plant to recognize that it's got to be strong itself in order to be able to deal with these bacteria. So the plant strengthens itself. Then there's a second kind of endophytic bacteria. That's just a word scientists use to describe bacteria that live part of their lives inside a plant and don't hurt the plant. So we've got these rhizophagy bacteria. Those are endophytic. But there are other bacteria that sneak into, say, for example, a stomata, or they sneak in to a place where there's branching, and they go into the plant and do all sorts of things because they produce phytohormones, auxins, gibberellic acids, and so they make the plant stronger. They may make the plant fruit earlier, give more fruit, give tastier fruit. We've discovered that there are bacteria that are inside the trichomes in tomatoes and cannabis. Yeah. Let's talk about cannabis for a second, that are literally being sprayed by the plant in order to protect itself from bacteria, because with cannabinoids, THC. Holy crow! We've got a whole different, and this whole idea of root hairs. So we've learned how to take care of the plant in a way that recognizes the soil food web. Now we've got this new thing, this Rhizophagy, and it's easy when you're talking generalized mulches, compost, to put the microbes back in the soil, to be regenerative and to add more to the soil to feed it. The whole concept is regenerative because you're not using poisons that your employees and your farm animals are being hurt with, blah, blah, blah. Now we've got these bacteria that we need to take care of in a slightly different way, and that will help us in slightly different ways. So for example, we already use some of these bacteria ourselves. Rhizobia, those nitrogen-fixing bacteria, those are endophytic bacteria. The plant causes them to be attracted. They fold root hair around them. They, create these bacteria. It's mycorrhizal fungi, same thing. Yep. They're attracted to the plant. They're feeding in the inside. Well, these bacteria can be added to the soil as well. And there are several that farmers around the world are using that are now trickling down into the grower situation, into the gardener situation. Azosporium. There's a bunch of them that are now coming to the market that the people are using. What we're learning from this whole concept of endophytic bacteria is that they ultimately end up in the seed coat.
Yes, yeah.
They go into the plant, they find their way to the flower, they end up in the seed coat, so that when that seed is planted the following season, that the right bacteria are there to support the plant in the manner that it's accustomed to being supported. They jump off the seed coat when there's germination. They populate the soil. They go back into the plant itself, et cetera, et cetera. So we've learned that we've got to be very careful about our seeds. No more sterilizing seeds. That's a very bad idea, because if you remove these bacteria, you don't get root hairs. Yeah. What? You and I were taught that root hairs are there to nourish the plant. They pick up nutrients. Yeah, that may be true, but they're also there, and they're formed by these bacteria, I say, in order to be shot back out into the soil again in new areas where they can regain nutrients and go back into the system again. So now when we start seeds in a paper towel, we know that the bacteria jumps off that seed coat into the soil, but if there's no soil but just a paper towel, we better use that paper towel. We now know that there is even more of a reason to leave the roots of a plant, if we're growing it in a container or in the ground or in the soil, and just cut it off at the ground level, take whatever you need, and then plant right there. There's bacteria and exudates that are in there that are going to attract the right kinds of bacteria. The right kind of bacteria is there if you're planting the same kind of plant. It's just, it all makes sense that we do these things in order to protect the microbes so that they can do what they need to do naturally. We don't have to help the microbes. We just need to protect them. And the way you protect them is to make sure that they've got enough food, that they've got the right environment, you're not disturbing them, either chemically or mechanically. All of these things are very, very easy to do. And once we do these things, we end up with less work ultimately for us and better plants for the world. Simple as that.
Yeah, it's exciting on multiple levels. One thing I like about it is encouraging people to not think of a plant as a single isolated organism in a vacuum, but rather you're dealing with a whole ecology within that, on and around and within that organism, and that you're growing a lot more than just a plant, but you're supporting a whole community of organisms that are working together to try to provide you with whatever you're after from that plant. And it's nice to anything that promotes that sort of systems level thinking, I get very excited about, because I think that's the kind of thinking we need to navigate a lot of the challenges that we're currently facing as
Right,
humans
because
on this planet.
if you just because if you just think about the plant alone, okay, I'm going to feed the plant a chemical. I'm going to give it water. You know, what you don't recognize is that by doing those things, it's good for the plant, maybe, but you're not getting soil structure because there aren't any bacteria there anymore. Which the slime creates the soil structure. You're scaring away the worms because they don't like the smell of the chemicals that you're putting there, and so you're not getting the porosity and the wormholes and the reservoirs, and there aren't worms to pull down the organic materials and stuff. Yeah, your plant's growing fine. You can put down, can I say it, Miracle Grow? Yeah, it works on the plant,
Right.
but it destroys the system. And yeah, that works if you want to be the system for the rest of your life. Who does? Where it doesn't work is when it begins to harm the environment, and not just you, but other people. And we end up with some serious problems. So, and particularly when you're talking about things like cannabis, which people are ingesting. I mean, if you're not doing it organically, you shouldn't be allowed to sell it, period. Yeah. It's just not good for you. You're ingesting this stuff, and it should be pure and clean, and grown the way nature intended it to do so. So, yeah, this is all really very, very, very interesting from the perspective of a system. That's what we really have. And even though I've got these separate books, they describe the system that operates in a beautiful geometric kind of way. It really is quite something. And this new stuff, the new photography, the pictures of these, oh my goodness gracious. And just as Dr. Elaine Ingham really developed the soil food web concept that we know and love today, Dr. James White is adding to it in a way that's equally as important. Plants can get up to 30%, so far that we know, and it may be even more, up to 30% of their nitrogen using the Rhizophagy cycle. That's a lot of nitrogen.
Yes.
And if it's getting it from bacteria, it doesn't need it from me and you. It doesn't need it from big combine, corporation that's destroying our soils, et cetera, et cetera. So this is all really good, exciting stuff, and it's a book people need to read. I changed in my mind the order that people should be reading the teaming series. The first one definitely teaming with microbes, but then teaming with bacteria. We didn't know about it when teaming with microbes came out. Then teaming with fungi, and finally teaming with nutrients. What happens to these nutrients once they're all in the plant? How do they get in the plant, and what happens once they get in the plant? But the fact that there are these really three modes of getting stuff into a plant is something that people need to know about. Mineral or soil mediated movement, where the bacteria and fungi get eaten by the protozoa and nematodes, and they poop into the soil. That stuff, the microbes put a charge on it, which is how they get into the plant, and it moves through the soil. That's soil mediated. Then we have the fungal mediated, which is the mycorrhizal situation. I always like to point out to people, because I'm a snob, you are not dealing with mycorrhizae fungi. You're dealing with mycorrhizal fungi that form a mycorrhiza.
Right.
And so when you see the word mycorrhiza, it's the root and the fungi. It's a mycorrhizal fungi. And then the third way is these bacterial endophytes, including the rhizobia and the rhizophagy. These are all so important to the way the plant operates, and getting to be more so.
Yeah.
One of the things about this book is, first of all, it was written 3 years ago, because that's how long it takes to get a book out. A lot of things we've discovered since then. I postulated based upon stuff that Dr. White was telling me about the trichomes and cannabinoids, et cetera, et cetera. Now there's been a paper published post publication of the book, of course, that shows that in those trichomes, in the core of the stalk, there are bacteria. The bacteria are just doing all sorts of great stuff. They're getting fed carbon by the plant in the trichome. They're using some of this carbon to create ethylene and nitrates, which get converted into nitro. I mean, it's just a beautiful system, and it's what people grow cannabis for. It's you
Can't escape it.
Yeah, it's in tomato plant trichomes. It's producing nitrogen, which the plant needs. And what we're discovering, or what Dr. White and people have discovered, is that every single plant with root hairs has these Rhizophagy bacteria. If you don't have them, you do not have root hairs. Wow. I mean, to me, at 73 years old, to get a new concept as to why we have root hairs
It's amazing.
is amazing. And some of the pictures in the book of the bacteria backing up inside the cell, they create ethylene, which creates that root hair, moving into the cell and getting blown out of the cell. That root hair may blow the tip off of it 5 or 6 times during its growth.
Yeah.
And each time it releases these bacteria out into the soil. So the end result is, when we learned about the soil food web from Dr. Eileen Ingham, we learned that plants were farmers. Mm-hmm. They put this exudate fertilizer out into the soil. They attract and grow bacteria and fungi who get eaten, et cetera, et cetera. We harvest the stuff the poop that's left over. What Dr. White's system shows us is that we're really dealing with ranching as well. We're bringing in the sheep, shearing off some of the wool, eating a couple of the lamb chops, and then putting the naked sheep back out so they can grow back the wool and get fatter, so that we can do it all again the next season. And so we're talking ranching and farming now. It's just unbelievable what plants are capable of doing.
Well, and that's, as you're describing all of this, what I keep thinking about is just how amazing it is that plants have evolved such a complex series of overlapping functions and systems, all in an effort, one, to maximize its fitness, but should things go awry, there are a lot of different partners and community members, neighbors, you could say, that the plant leans on for support to get through the stressful times. And I find that complexity just really brilliant.
Yeah, it is amazing. So some of these endophytic bacteria are I always pronounce this wrong, cytophores, I guess it's cytophores. I want pores. They go out and they cleate metals. They tie them up in the soil.
Yeah.
And sometimes they then let them migrate into the plant if the plant needs them. But very often they do it so that the plant won't get poisoned by some of these metals. Too much arsenic and blah, blah, blah.
Basically fixing them a bit.
Yeah. Cytophores. That's SIDER. Oh, God, PHORES. Okay, anyway. It's in the book, folks.
We will find it,
definitely want to get teaming. You want to get teaming with bacteria for no other reason to see the pictures there.
They really are amazing. I was just sitting here flipping through to see. I marked a lot of different pages, and I'm trying to find some of them that you mentioned. Like one that's here is just a close up of the root hairs, where you can actually. I don't know how well this will actually come up on the video, but I love that picture.
And if you look carefully at that picture, and you can see that those root hairs are full of bacteria. How did we not know that?
Right.
Well, we didn't know it because we didn't have that kind of microscopy available.
Yeah. And it highlights, the better our technology gets, the better our tools are for looking closer at things. You just never know what we will see that just changes your entire perspective of something you thought you knew so well already.
Right. Or how you would use that technology. So, for example, I postulate in the book, and I think I'm right. We already have a database now that has all of the microbes. Well, let's start with bacteria. All of the bacteria that you'll find in soil. Basically, maybe there's some that we haven't discovered yet. But that database exists. You can take a sample of soil and send it in, and if you want to spend the money, and they'll tell you what bacteria you have in your soil. Well, that's coming to a phone near you, no question in my mind. Where you're going to be able to take a picture of a sample of soil that's been in water or something like that, and get back an answer as to what's in it. It's coming, and it's going to be really exciting when it does. It's amazing what science will do. And, having gone through this whole COVID period where there was a lot of anti-science, et cetera, I only hope we don't end up with somebody saying, no, this is all BS and don't buy these books because it's not true.
Just monocrop everything, keep using your fertilizers and proceed.
Yeah, keep using chemicals. Rototillers are fabulous. You can do it every day. Yeah, but the science behind this is irrefutable.
Yes.
And that's the beauty of it. And as gardeners, for too long, this generation and my generation of gardeners have been doing too many things based upon chemical nitrogen, artificial fertilizers, an angry Scotsman yelling at us every spring that we needed to be doing stuff, as opposed to teaming with microbes. It's just simple as that.
It is, and one thing I wanted to highlight, because I don't know if we broke it down completely, but I wanted to, for the nerds out there that really want to get into some of the biochemistry of this, one thing I'm fascinated by is this sort of series of reactions that happens when the bacteria enters the roots. The plant has this reaction of releasing reactive oxygen species that help to get that wall of the bacteria off and start to break it down. The bacteria starts to freak out a little bit, starts to produce ethylene.
Right.
And you start to get also nitric oxide produced as well as kind of a defensive mechanism.
Right. The bacteria get trapped in there. So what happens is the bacteria are sitting out there in the soil, happily eating away. They're in a slime.
Yes.
Up against the wall, and all of a sudden they smell. I'm being anthropomorphic here. They smell butter. They smell popcorn, and they move towards that popcorn because it tells them in their language that there's an empty house they can go live in and don't have to compete with all the other jerks in the slime that they live in there. And as they move into this new house, they end up going through the cell wall. They're very thin because they're very brand new, of a meristem cell in the root. They find themselves through the cell wall, and the plant itself then reacts by spraying the bacteria with a super oxygenated material, super oxygen, ROS, ROS, and it does. It pulls off the bacteria's cell wall, which contains, in particular, a little bit of nitrogen, but it's got some of the minor nutrients and metals and things like that, depending on whether it's gram positive or gram negative. And then two things happen. First, the plant says, I don't want this spray to remove my wall, so I better strengthen myself. And the second thing happens is that these bacteria produce the ethylene. They're always producing the ethylene. They're using one of the exudates to produce that ethylene, and that causes the cell wall of the plant to grow. And as it grows, it leaks out little bits of carbon, which feed the bacteria and keep them alive. The other thing the ethylene does is it enables the plant, the bacteria, to create a spray that acts as an antioxidant that weakens the spray that took off their cell wall, and so you get this neutralization. You don't have cell walls on these things, and there's cyclosis going on around at the same time. And so the bacteria multiply, because they do so every 20 minutes anyway. Usually they form these quads. They move around this space, continually producing ethylene, continually causing the root cell to grow, release carbon so that the bacteria stay alive because they need the carbon, and cause the bacteria to produce the nitrite, which turns to nitrate and feeds the plant. This goes on for about 2 days. And by then you start to get so the bacteria are basically trapped. If they stop producing the ethylene, they don't get the carbon, they die. And if they don't produce the nitrate, they die because they get killed by the spray that's coming from the root cell. So for 2 days this goes on. Eventually they get caught up in the cell space, in the a little bump in the cell wall. The ethylene doesn't circulate quite as much. You don't get growth of the cell entirely. You just get growth of this little tube that comes out of the cell, which is the root hair. And then they go out. It's just fabulous. Beautiful, beautiful chemistry. And the plant is in control. The plant is the boss, just like it is with the ectomycorrhizal and endomycorrhizal, just like it is with the rhizobia. It's the plant that sends out the signal. Now, why would a bacteria, take that signal? Because they can get food and space to live without competition from other guys, from the bad guys, et cetera, et cetera, et cetera. It's just a beautiful, beautiful system. And I'm so glad that we've now got a bigger picture of the soil food web. It's just beautiful, beautiful, beautiful.
I mean, it's amazing to even be able to visualize all of this to the detail we can, and even on a molecular level. The fact that we've gotten this far is really incredible, and I have to ask you because I feel like it's just begging to happen. Especially now that you've got in your mind kind of among your 4 books the best order for people to read them and everything. I have to ask, is there a Teeming with Life that's coming in the future that's just one book with everything combined together?
I don't think so. I don't know. I mean, I always tell my wife, I just can't write another. I have one book out on cannabis, autoflowering cannabis, so that's 5 books. Just as an aside, to write a book, a couple of things. First of all, you got to come up with an idea that interests you the writer.
That keeps you passionate.
And that you think might be worthwhile to interest other people. And then you have to go down into this little teeny rabbit hole when you've got everything laid out and organized, and you don't want to come out of there very often. And so, it's dinner time. Oh my God, cute. I mean, it's just, and for a year, you're in this little rabbit hole, and then you send it into the editor, and you just keep your fingers crossed. I hope they understand what you're writing. Yeah, so I don't know if there's another one. I'd love to write a novel, but I'm not that good. I could see some other ones here, but oh my God. And it's not just the writing, you have to get pictures.
Yeah, formatting, it's all yes. Yeah.
Well, the press does all the formatting, but you got to get the pictures.
Yeah.
Whew, not easy. You think it's easy, but it's not.
Well, and you have to get the permissions for everything. Yeah.
Yeah. So anyway, I'm not that doing a podcast is any easier by any means, particularly when you have to do lots of them and frequently. But yeah, writing a book, I don't know if I got another one in me. I keep wondering about viruses in soil. Oh my God.
That's a big one. Yeah. That started to come up a lot, especially in the cannabis world. Yeah. There's a lot of interest in the viruses, especially the hop latent viroid that folks are dealing with and stuff. There's a lot of questions people are asking. I could see that. Yeah.
Yeah, and I can see a book on convincing people that we need to change our education system to concentrate on microbes instead of dinosaurs.
Yeah.
That's
the
bottom
things that
line.
we're actually interacting with on a regular basis that affect everything we do.
Exactly, and if little kids can learn how to pronounce those dinosaur names, they can learn bacteria and fungi and
they absolutely
nematodes and protozoa.
They absolutely can. My daughter is 3 years old, and I talk to her as if she's in college sometimes because I'm always working on different projects, experiments, and things, and I just tell her the big words, molecule names, and stuff. My big pride is she recognizes plants. So we
Yeah.
were in the Pacific Northwest last week, and we're hiking around, and she looked up and she said, "Whoa, that's a tall tree. That must be a redwood." And
Oh yeah.
just
Yeah.
see my 3-year-old like
Yeah.
trying to recognize plants, and
Right.
she's like, "I think that's
Right.
calendula." I'm like, "Man, you're making me so proud right now."
Yeah.
So
There you go.
they will learn if you
Yeah.
just tell it to them and keep presenting it to them. They remember, they absorb, and I totally agree because my background was in science education before I got into more like media, science education that way. And I think the whole way we handle education needs to be changed, but specifically science education.
Yeah.
It's so obvious that there's a huge problem, a huge deficit in public scientific literacy, and it's scary because it affects all of us
Yeah.
in a big way.
Right. Right. Yeah. And I always tell people the redwood's a great example. I mean, the biggest organisms on the earth are supported by the littlest organisms,
right.
and we're all working together. And what happens in the soil happens in your stomach, including this bacterial situation. The same system works. They back into the intestines the same way they back into the cell. Same chemical. It's mind-boggling. It's really, really mind-boggling. So I really encourage people, not because I wrote them, but because they will make you a better grower, gardener. You should get the teaming series. If it doesn't open your eyes, I'll be very surprised. Very surprised.
I completely agree. And as someone who is a gardener and I discovered your books when they first came out and have been following them, I can echo that from a relatively, I guess I'm biased now, but at the time I was not biased when I discovered the books. And I definitely love them. They're, just dense enough, just long enough to give you a lot to digest and think about, but it's really written for anyone. A lot of fairly complex scientific concepts that are broken down in a way that just about anybody can understand, which is something I always really appreciate when I find an author that's able to do that, to bridge these worlds that often have such a large gap between them, to make people not so
the reason
scared.
why, yeah, I do it because I'm not that bright, and I gotta dumb this stuff down so that I can understand it, and if I can understand it, anybody can understand it, and so that's how they get that way. It's nothing brilliant on my part by any means, and I always like to tell people when we do these interviews, my hat is off to Dr. Elaine Ingham,
Yes.
Dr. James White. I did not invent any of this stuff. It's not my research, and I thank goodness that they did it and their students did it and worked it all out, continue to do it, so that you and I can get the benefits of it. But this is not my work. What I'm trying to convince people is to read the work of others,
Yes.
basically.
You're the bridge. You're the bridge that gets
I
people
guess so.
to, all of that deeper science and everything that's behind that. You're the messenger shouting, look at this amazing stuff that is going on. And I really, really appreciate it a lot, especially from one science educator to another. I think it's really important work, and definitely everyone listening, especially if you're growing things, but even if you're just trying to understand how nature works, because this is all applicable in ways that far exceed gardening and that sort of thing. Definitely pick up the teaming series, and to go over your recommendations. You said start with teaming with microbes if they're starting out, then teaming with bacteria, then teaming with fungi, and teaming with nutrients, and you'll get all the way from the soil into the plant and back again, there and back again.
Right.
So this is great, Jeff. Thanks so much for being willing to come on today and to share all of this with us. I hope that folks listening are now more excited to dive into learning about bacteria. I think it's something, microbiology in general, it gets underappreciated, and it deserves a lot of respect, and there's a lot of really cool stuff going on under the surface. And so yeah, thanks for coming on. Thanks for spurring our curiosity and spending the time to talk with me today. I really appreciate it.
My pleasure. Let's do it again.
Absolutely. I will definitely hit you up for that. And for folks that want to learn more about you or finding the books, do you have a personal website to direct people to or anything like that?
Yeah, I never do anything on that website. I think Twitter handle Gardner Jeff at Gardner Jeff is probably the easiest
Nice,
way.
at Gardner Jeff.
Yeah, I'm too old to work the damn website all the time.
Well, and everything, like we were mentioning, I was running into technological problems just trying to get this interview going, and everything seems to change so fast these days. You get used to one service, and you have to adjust to another one a year later. So I'm about there too. So, well, thanks so much.
Yep.
Have a great rest of your day, and everyone, look up Jeff Lowenfels, look up Teaming with Bacteria, and the whole teaming series. There's 4 books. Check them all out. They're all amazing, and hope you enjoy them as much as I do. And with that, everybody.
Yeah, and don't forget that it's teaming. TEA.
Good clarification, not TEE. Yeah, TEA. Teaming, partnering with, teaming with. Yes. Right. Great clarification. Very perfect for audio. Yeah, that's the little things we often forget. Yeah, if you're searching for that, teaming as if partnering, TEAM, teaming with microbes, teaming with bacteria, teaming with fungi, and teaming with nutrients. And yeah, this has been great. Thanks so much, Jeff, and we will definitely catch up with you again soon.
Great. Thank you very much.
All right, everybody, stay curious and take it easy.